Oligonucleotides and therapeutic small interference RNA, or siRNA, hold great promise as novel therapeutic agents designed to specifically and selectively inhibit the production of disease-related products, with Onpattro or patisiran being the first approved siRNA drug product in 2018. Oral bioavailability is generally very low, ranging from 1 to 3%. After intravenous administration, oligonucleotides follow generally two-compartment characteristics and are rapidly cleared from plasma, predominantly via distribution processes with a half-life of 0.5 to 1 h depending on the dose. The plasma pharmacokinetics of various oligonucleotides are generally independent of their sequence and chemistry, as plasma clearance is primarily determined by distribution processes. After intravenous administration, oligonucleotides are detected in nearly all tissues and organs except for the brain and testes, suggesting significant transport barriers in these tissues. The extent of tissue uptake is dependent on the dose amount as well as dose rate. Major accumulation of oligonucleotides occurs in liver and kidneys, and to a lesser extent in spleen, bone marrow, and lymph nodes, which seems to be independent of oligonucleotide sequence. Chemical modification of the backbone structure, however, may alter protein binding and organ distribution. The mechanisms for uptake into target cells have not been fully elucidated yet, but these processes are energy, temperature, and time dependent, and include most likely pinocytosis and podocytosis. Oligonucleotides are cleared from tissues by nuclease-mediated metabolism, with half-lives that vary between 20 and 120 hours, depending on the organ or tissue. Successive removal of bases from the 3' end is the major metabolic pathway in plasma, while both 3' and 5' exonuclease excision may occur in tissues. Exonuclease metabolism in plasma and tissues is rapid, with 30 to 40% of oligonucleotides having at least one nucleotide removed after 5 minutes in plasma. Endonuclease-mediated degradation of oligonucleotides is generally not observed. Oligonucleotides are highly bound to plasma proteins that protect them from renal filtration. Urinary excretion is a major route of excretion for oligonucleotides, regardless of sequence or chemical structure, with the majority being shorter-length metabolites rather than unchanged parent drug. Urinary excretion is nonlinear, with a greater fraction excreted at higher doses. Potential mechanisms include saturation of plasma protein binding, as well as tubular reuptake mechanisms. Only a minor fraction of the dose is excreted into feces, although enterohepatic recirculation has been suggested. In comparison to peptides, proteins, and oligonucleotides, much less is known about the pharmacokinetics of recombinant plasmid DNA used as a drug in the novel treatment approach of gene therapy. The in vivo disposition of plasmid DNA and its complexes depends largely on its physicochemical characteristics, a strong negative charge, and high molecular weight. After intravenous administration in rats, plasmid DNA is detected in all major organs including lungs, liver, kidney, and spleen. Low-level detection in the brain is most likely an artifact from residual blood, given that plasmid DNA is unlikely to cross the blood-brain barrier. After intravenous administration in mice, plasmid DNA is rapidly eliminated from the plasma due to extensive uptake into the liver as well as rapid degradation by nucleases, with hepatic uptake clearance approaching liver plasma flow. Plasmid DNA is preferentially taken up by liver nonparenchymal cells, such as Kupffer and endothelial cells via receptor-mediated processes. Analysis of the functional forms of plasmid DNA in rats revealed that supercoiled plasmid DNA rapidly disappears from plasma with a half-life of 0.15 minute. Approximately 60% of supercoiled plasmid DNA is degraded to open circular plasmid DNA, which is subsequently nearly completely converted to linear plasmid DNA. Conversion of open circular to linear plasmid DNA followed Michaelis-Menten kinetics, while linear plasmid DNA was removed with a half-life of 2.1 minute. The slower elimination of open circular and linear plasmid DNA compared to supercoiled plasmid DNA was suggested to be related to a stronger interaction with plasma macromolecules that might offer some protection from plasma nuclease degradation. In summary, Biotech drugs underlie the same pharmacokinetic and pharmacodynamic principles as traditional small molecule drugs. On the basis of their similarity to endogenous compounds or nutrients, however, numerous caveats and pitfalls related to bioanalytics and pharmacokinetics have to be considered and addressed during the development process, and may require additional resources. Furthermore, pharmacodynamics is frequently complicated owing to close interaction with endogenous substances and specific feedback mechanisms. Biotech drugs, including peptides, proteins and antibodies, oligonucleotides, and DNA, are projected to cover a substantial market share in the healthcare systems of the future. It will be crucial for their widespread application in pharmacotherapy, however, that their respective drug development programs are successfully completed in a rapid, cost-efficient, and goal-oriented manner. A more widespread application of pharmacokinetic and pharmacodynamic concepts, including exposure-response correlations, has repeatedly been promoted by industry, academia, and regulatory authorities for all preclinical and clinical phases of drug development, and is believed to result in a scientifically driven, evidence-based, more focused, and accelerated drug product development process. Thus, pharmacokinetics and pharmacodynamics concepts are likely to continue and expand their role as a decisive factor in the successful development of biotechnologically derived drug products in the future.